The forgotten truth behind the most important scientific discovery. What if I told you the scientist who discovered penicillin, the miracle drug that has saved over 200 million lives, actually tried to abandon his finding, and that the real heroes who turned it into medicine have been virtually erased from history? September 1928. Alexander Fleming returned to his laboratory at St. Mary's Hospital in London after a 2-week holiday in Scotland. A notoriously untidy researcher, he'd left several petri dishes of Staphylococcus bacteria scattered across his workbench. What he found upon his return would change the world, but not in the way we've been taught. One dish had been contaminated by a strange mold, and around this fuzzy invader, the deadly bacteria had simply disappeared. A clear ring surrounded the fungal colony, as if something was actively destroying the germs. Fleming's trained eye recognized something extraordinary. He isolated the mold, identified it as Penicillium notatum, and discovered it produced a substance that killed a range of harmful bacteria without damaging human cells. He called it penicillin. This is where the story usually ends in textbooks, with Fleming's brilliant observation earning him the Nobel Prize and global fame. Schools teach this as a tale of scientific genius, of one man's keen eye catching what others might have missed. But this narrative omits something crucial. Fleming gave up on penicillin. The complete story reveals something far more profound about how scientific breakthroughs actually happen. It's a tale of refugees and war, of women whose names were never recorded, of a moldy cantaloupe that saved millions, and of the uncomfortable truth that our greatest medical triumph came dangerously close to dying in obscurity. The discovery that almost died. Fleming published his findings in 1929, but his paper generated barely a ripple of interest in the scientific community. More critically, Fleming himself couldn't see how to make his discovery useful. The mold juice, as he called it, was extraordinarily difficult to produce in meaningful quantities. It was unstable, losing potency within days. His attempts to purify it failed repeatedly. The technical challenges were immense. Penicillin degraded when exposed to heat, light, or even slight changes in acidity. Fleming's crude extraction methods yielded a brownish liquid that was mostly impurities. He had no way to concentrate or preserve it. When he tried to inject it into animals, he couldn't produce enough to see meaningful effects. By 1931, Fleming had essentially abandoned penicillin research. He continued to use it occasionally in his lab to selectively grow certain bacteria, a niche application that helped no one fighting infection. For a decade, penicillin sat forgotten in scientific literature, a curiosity with no practical application. Meanwhile, people continued dying from simple infections. The pre-antibiotic world was a place of routine horror. A scraped knee could become a death sentence. Pneumonia killed 30% of those infected. Childbirth fever claimed thousands of new mothers annually. Syphilis ravaged bodies and minds. Surgery was dangerous, not because of the procedure itself, but because post-operative infections killed patients who should have survived. Fleming had discovered penicillin, but he had not created a medicine. That transformation would take an unlikely team of scientists working under the shadow of war to accomplish what seemed impossible. Enter the unlikely heroes. In 1938, an Ernst Boris Chain arrived at Oxford University's Sir William Dunn School of Pathology. Chain had fled Nazi Germany in 1933, leaving behind his family, his career, and his homeland. His mother and sister would later perish in the Holocaust. In England, he found himself working under Howard Florey, an Australian-born pathologist with a gruff demeanor and an obsessive interest in bacteria-killing substances. Florey was brilliant, demanding, and difficult. Born in Adelaide, he'd risen through British academia through sheer determination. He rarely smiled, worked punishing hours, and expected the same from his team. But he had vision. He believed natural antibacterial agents held the key to fighting infection. Florey and Chain were studying natural antibacterial agents when they stumbled across Fleming's old paper on penicillin. They decided to investigate whether this old mold might have potential. Neither had any intention of making medical history. They were simply following scientific curiosity, funded by a small Rockefeller Foundation grant. What they discovered was daunting. Extracting even tiny amounts of penicillin from mold cultures was monstrously difficult. The substance degraded almost as fast as they could produce it. After months of work, they'd managed to create only enough penicillin to fill a few small vials. But Chain was a skilled biochemist, and he was determined. Working with Norman Heatley, a biochemist with a genius for improvisation, they developed a complex freeze-drying technique to concentrate the penicillin. Heatley was the team's secret weapon, a quiet man who could build sophisticated equipment from basic materials. He created elaborate systems of glass vessels, rubber tubing, and carefully controlled temperatures. Heatley's contributions were remarkable. He invented a method to extract penicillin using back extraction between water and organic solvents, dramatically improving yields. He designed specialized vessels for growing mold cultures and developed an assay method to quickly test penicillin potency. Without Heatley's practical genius, the Oxford team would have failed. By early 1940, they'd produced enough penicillin to test on mice. The experiment was simple but dramatic. They injected 8 mice with lethal doses of Streptococcus bacteria. 4 mice were received penicillin. 4 did not. By morning, the 4 untreated mice were dead. The 4 treated mice were alive and healthy, scampering around their cage as if nothing had happened. Fleming's assistant watching the surviving mice reportedly said, "It looks like a miracle." The team published their results in The Lancet in August 1940. Unlike Fleming's paper 11 years earlier, this one made clear that penicillin could be practical medicine. But now they faced the nearly impossible task of producing enough penicillin to test on humans.
The first human life. In 1941, as German bombs fell on London during the Blitz, the Oxford team worked frantically to scale up production. The challenges were staggering. To produce enough penicillin for a single human treatment required hundreds of liters of mold culture growing over weeks. They commandeered every available container in the laboratory. They grew mold in bedpans, hospital containers, pie dishes, and ceramic pots. They filled an entire room with vessels containing the golden liquid. The laboratory smelled strongly of the mold cultures. Even with this desperate expansion, they could produce only tiny amounts of penicillin. The team worked around the clock. Lab assistants monitored cultures through the night. Chain and Heatley developed ever more efficient extraction methods. Florey coordinated the operation like a general managing a campaign. Their first patient was Albert Alexander, a 43-year-old police constable. Alexander had scratched his face on a rosebush while gardening. The small wound had become infected, and the infection had spread catastrophically. By the time he reached Radcliffe Infirmary in Oxford, the infection had consumed his face. His eyes were swollen shut, his scalp was oozing pus, and his lungs were filling with abscesses. The doctors had already removed one of his eyes, but the infection continued spreading. He was dying. On February 12, 1941, Florey administered the first dose of penicillin. The effect was dramatic. Within 24 hours, Alexander's fever dropped. His appetite returned. The swelling in his face began to subside. After 5 days of treatment, he was sitting up in bed, eating meals, and talking with nurses. It appeared to be the miracle everyone had hoped for. Then, the penicillin ran out. The Oxford team had used every drop they possessed. They'd even collected Alexander's urine and extracted the penicillin his body had excreted, purifying and reusing it. But it wasn't enough. Without continued treatment, the infection roared back. Albert Alexander died on March 15, 1941. It was a devastating failure and an extraordinary proof of concept. Penicillin worked magnificently. The team simply needed more of it. Much more. They treated 4 more patients that spring with their limited supplies. All 4 survived. The case that proved penicillin's potential most dramatically involved a 15-year-old boy with an infected hip who was expected to die within days. After 6 weeks of penicillin treatment, he recovered completely. The industrial challenge. This is where the story becomes truly surprising. Britain, in 1941, was fighting for survival. German U-boats prowled the Atlantic. Factories were being bombed nightly. Resources were desperately scarce. British pharmaceutical companies, focused on producing medicines for the immediate war effort, couldn't spare the capacity to develop penicillin production. Florey made a fateful decision. He would take his research to America. In July 1941, Florey and Heatley flew to the United States with a small amount of penicillin and the secret to producing it. They'd smeared the precious penicillium mold inside their coat pockets, reasoning that if the plane crashed or was shot down, at least one of them might survive with the mold intact. They landed in Lisbon, then flew to New York. America wasn't yet at war, but it had industrial capacity Britain lacked. Florey hoped American pharmaceutical companies could solve the production problem. He was initially disappointed. Most companies were skeptical. Penicillin seemed too fragile, too difficult to produce commercially. Then Florey met with the U.S. Department of Agriculture's Northern Regional Research Laboratory in Peoria, Illinois. The scientists there specialized in fermentation, growing microorganisms at industrial scale. Peoria seemed unlikely for a medical revolution. A small city in the American Midwest, it was far from prestigious East Coast universities. But the laboratory there had exactly what Florey needed: practical knowledge of growing microorganisms in large quantities. What happened next was revolutionary, though it receives almost no attention in popular histories. The unsung innovation. A laboratory assistant in Peoria named Mary Hunt changed everything. Hunt's job was to scout local markets for mold samples, hoping to find a strain of penicillium that produced more penicillin than Fleming's original mold. The logic was simple. Fleming had found one mold by accident. Perhaps a more productive strain existed in nature. For weeks, Hunt brought in moldy fruits, vegetables, and breads. The team tested each sample, measuring penicillin production. Most produced nothing. Some produced tiny amounts. None exceeded Fleming's original strain. Then one day, in 1943, Hunt arrived with a moldy cantaloupe from a local market. The mold growing on this ordinary piece of fruit turned out to be a strain of Penicillium chisogenum that produced more than 1,000 times the penicillin of Fleming's original mold. It's difficult to overstate how important this discovery was. This single cantaloupe mold became the ancestor of virtually all penicillin-producing molds used today. Every dose of penicillin administered since has descended from Mary Hunt's cantaloupe. She earned the nickname Moldy Mary, but her contribution is rarely mentioned outside specialized histories. Meanwhile, other Peoria scientists solved the production challenge through fermentation engineering. They discovered that adding corn steep liquor, a waste product from corn processing, dramatically increased penicillin yields. They developed deep tank fermentation methods that could produce penicillin at industrial scale. The transformation was remarkable. In early 1942, the U.S. had produced only enough penicillin to treat a few dozen patients. By the end of 1942, American companies were producing enough to treat 100 patients. By D-Day in June 1944, they produced enough for every Allied soldier who needed it. By the war's end, U.S. companies were producing 650 billion units of penicillin monthly. This achievement was as significant as radar or the atomic bomb, yet the scientists who accomplished it remain virtually unknown. While Fleming became world famous, the Peoria researchers who made penicillin practical received little recognition.
The forgotten women. The industrial scale-up also relied heavily on women scientists and technicians, another forgotten part of the story. During World War II, as men enlisted or were drafted, women filled crucial roles in penicillin research and production. Margaret Pittman, working at the National Institutes of Health, standardized penicillin testing methods, ensuring consistent potency across different manufacturers. Dorothy Hodgkin used X-ray crystallography to determine penicillin's molecular structure, work that would later earn her the Nobel Prize in Chemistry in 1964, though she's rarely mentioned in penicillin histories. Hundreds of female technicians ran the fermentation tanks, monitored mold growth, and conducted quality control tests. One facility employed over 500 women in penicillin production. Their meticulous work ensured that soldiers received medicine that would save their lives. Yet photographs and newsreels of the era focused almost exclusively on male scientists and executives. These women were often paid less than their male counterparts and received little credit for their contributions. After the war, many were pushed out of their positions as men returned from military service. Their names weren't recorded in official histories. Fleming's return to glory. Meanwhile, Alexander Fleming had become famous for doing relatively little after his initial discovery. The British press celebrated him as the discoverer of penicillin, largely ignoring Florey, Chain, and the American teams. Fleming gave speeches, received honors, and accepted accolades. He was knighted in 1944. Florey, by contrast, actively avoided publicity. He believed scientists should work quietly and let their results speak. This modesty cost him public recognition. When people thought of penicillin, they thought of Fleming. The tension between Fleming and the Oxford team was palpable. Chain, in particular, felt bitter about Fleming receiving credit for work he hadn't done. Fleming had discovered the mold. Florey, Chain, and Heatley had created the medicine. These were different achievements, yet the public conflated them. In 1945, Fleming, Florey, and Chain shared the Nobel Prize in Physiology or Medicine. Heatley, whose experimental genius had been crucial to purifying penicillin, was excluded. The Nobel Committee's rules allowed only 3 recipients, and Heatley's contribution was deemed less significant than Chain's biochemistry and Florey's leadership. Heatley received belated recognition in 1990 when Oxford University awarded him an honorary doctorate, the first such honor the university had granted in 800 years. He was 81 years old. The woman who'd found the cantaloupe never received any formal recognition. The real miracle. Here's what makes this story so surprising. The discovery of penicillin wasn't a miracle of individual genius. It was a triumph of collaboration, persistence, and industrial innovation spread across 2 continents and dozens of institutions. Fleming deserves credit for recognizing something unusual, but recognition isn't invention. The real heroes were the people who refused to let the discovery die. Florey and Chain, who saw potential in a forgotten paper. Heatley, who designed the extraction methods. Mary Hunt, who found a better mold on a cantaloupe. The Peoria scientists who solved fermentation at scale. The pharmaceutical companies that retooled factories. The hundreds of technicians, mostly women, who produced billions of units.
the military doctors who administered it to wounded soldiers. By the war's end, penicillin had treated everything from battlefield wounds to syphilis, from pneumonia to scarlet fever. It reduced mortality from bacterial infection by 90% or more. Diseases that had killed for millennia became curable. Children stopped dying from ear infections. Women survived childbirth complications. Simple surgeries became safe. The impact was so profound that life expectancy in developed countries jumped by nearly a decade in the post-war period. Penicillin and subsequent antibiotics contributed more to human longevity than any other medical intervention in history. The uncomfortable truth. But here's the uncomfortable truth that makes this story even more complex. Fleming recognized penicillin's potential danger from the beginning. In his 1945 Nobel Prize acceptance speech, Fleming warned, "The time may come when penicillin can be bought by anyone in the shops. Then there is the danger that the ignorant man may easily underdose himself, and by exposing his microbes to non-lethal quantities of the drug, make them resistant." He was precisely right. By the 1950s, penicillin-resistant bacteria were emerging. Today, antibiotic resistance threatens to return us to a pre-penicillin world where simple infections can kill. The World Health Organization calls antibiotic resistance one of the greatest threats to global health. Fleming discovered penicillin through a contaminated petri dish, a fortunate accident. But the crisis of antibiotic resistance emerged from human choices. Overprescribing antibiotics, using them in agriculture, patients not completing treatment courses, and pharmaceutical companies abandoning antibiotic research as unprofitable. The forgotten truth behind the most important scientific discovery is that it was never just one person's achievement. It was the product of an entire generation working urgently under wartime pressure, pooling knowledge across borders and scaling production through industrial innovation. Legacy and lessons. Today, when we take antibiotics for granted, we forget the thousands of people who made them possible. We remember Fleming's name, but not Mary Hunt's Cantaloupe, not Heatley's equipment, not the female technicians monitoring fermentation tanks. This forgetting matters. It reinforces the myth of the lone genius when science is almost always collaborative. It erases the contributions of women, refugees, and technicians whose work was essential. And it obscures how scientific discoveries become medical realities through engineering, manufacturing, and the unglamorous work of scale-up and production. The real miracle of penicillin wasn't Fleming's observation. It was that disparate teams across the world, biochemists in Oxford, fermentation experts in Peoria, pharmaceutical companies in America, military doctors in field hospitals, coordinated to transform a laboratory curiosity into a weapon against humanity's oldest enemy. Ernst Boris Chain, the refugee whose biochemistry made penicillin a medicine, died in 1979. Howard Florey died in 1968. Norman Heatley died in 2004. Mary Hunt died in 1966. Their names aren't taught in schools. Their faces don't appear in documentaries. Alexander Fleming died in 1955, celebrated worldwide as the man who discovered penicillin. He earned that recognition, but not alone and not for the whole story. The forgotten truth is this: the most important scientific discovery in history succeeded because thousands of people, many of them forgotten, saw a good idea and refused to let it die. They worked without glory, many without recognition, to turn one man's observation into millions of lives saved. That collaborative triumph, more than any individual brilliance, deserves to be remembered.